Hello students, welcome to our online video tutorial class. So I think you have seen the previous video. In the previous video we have discussed about the kingdom Protista. We have discussed there about the special features present in case of those organisms of kingdom Protista, as well as we have discussed about their classification entirely. So I have decided that in this specific video I will be discussing about the kingdom Fungi. In the kingdom Fungi, first of all, we all know that the entire basic features of kingdom Fungi we have already studied in the initial of the chapter. That means when I have talked about the introduction, on that portion I have discussed about that part. So in this portion we will be discussing about the special features as well as the classification of kingdom Fungi like the kingdom Protista. So let's see. First, the special features we will be discussing. In the special feature, first of all, the feature is coming, that is the mode of reproduction. In the mode of reproduction, you all can see that reproduction commonly in case of fungi can be of two types. One is asexual mode of reproduction, another one is sexual mode of reproduction. So in case of this asexual mode of reproduction, whatever the different structures can be present, let's see. In case of asexual mode of reproduction, there are three different types of spores are commonly found, or spores can commonly present. First one, that is zoospore. Zoospore means, from the name we can understand, this is the flagellated spore, and that's why this spore can show the motility. It can move from one place to the another place. So zoospore will be the motile flagellated spore which is helping the fungi organism during their asexual mode of reproduction. Conidia. Conidia is another mode of the reproduction where you can see Conidia is the non-flagellated, non-motile spore, and by this specific spore, obviously, the fungi organism can reproduce, they can perform the asexual mode of reproduction, but this spore can't move from one place to another place like the zoospore. Okay, can't move because of due to absence of the flagella. Third one, you can see here another type of the spore it is mentioned, that is aplanospore. In the aplanospore, we all know that this is also another non-motile spore. It is also non-flagellated like the Conidia. But one thing we have to keep it in your mind, or we have to keep it in our mind, that aplanospore and Conidia, there is a basic difference. What is the difference? Conidia is commonly containing the thick walled, and aplanospore is commonly thin walled. So based on the thickness of the cell wall, this Conidia and aplanospore both are actually different from each other. There is no other difference, structural little bit difference is present. And always keep it in your mind, Conidia type of the spore, it is commonly found in case of penicillium. Except these three kind of spore formation, there is one more asexual mode of reproduction in case of fungi, that is vegetative propagation. And in case of vegetative propagation, fungi are commonly showing only one kind of vegetative propagation, that is budding. We all know budding is commonly shown by the yeast. So budding can be another type of the asexual reproduction, or rather you can say vegetative mode of propagation or vegetative reproduction. Going for the next slide. In the next slide you can see properly the three kind of the spore structures shown. First one, that is the zoospore. In the zoospore structure you can see that it is containing the motile spore. Okay, motile flagella is there, and for that reason it can show the motility. In case of Conidia, you can see these are the small pores which are commonly known as Conidia, and these are actually growing within some sac-like structure which are conidiophores, or these pore sacs are commonly known as conidiophores. And another type of the spore you can see here, that is the aplanospore. This is also non-flagellated spore like the Conidia, and this aplanospore is mainly how it is different from the Conidia, already I have told. This is actually, the wall is commonly thin, but in case of Conidia the wall is comparatively thicker than the aplanospore. So this is the only difference. Going for the next slide. In this next slide you can discuss about the sexual mode of reproduction. In the previous slide we have discussed about the asexual mode. Here, first of all, when we will be start discussing about the sexual mode of reproduction, we have to know whatever the various structures are actually involved in this reproduction. So in this reproduction also some specific spores are involved, but maybe the spore word is present behind each and every name, but these spores are actually not structurally spore like. They are showing the function like gametes, how the gametes are performing in the sexual mode of reproduction, or whatever the fusion or fertilization is happening, that same phenomena, that same specific type of function is shown by this kind of spore also. Because already I have told, maybe the spore word is there, but functioning they are all like gametes. So first of all, in case of ascospore, you can see these are the sexual spore, which is commonly found in case of one kind of fungi, that is the ascomycetes. In the next slide we will be discussing about this ascomycetes. And keep it in your mind, this ascomycetes fungi, in case of that spore is generally growing within a fruiting body, that is a reproductive structure in case of fungi, that is commonly known as ascus. In case of ascomycetes fungi, the spore is ascospore, and within which the spores are generally produced, that is the fruiting body, that is ascus. Next, basidiospore. From the name we can understand this spore is commonly produced by another class of the fungi, that is Basidiomycetes, and in case of them they have the fruiting body, that is basidium, and due to presence of that basidium, the basidiospore is commonly growing within that basidium. Okay. Third one, oospore. Oospore is commonly found in case of another class of the fungi, that is Oomycetes. Oomycetes is the another class of fungi where the oospore is generally forming, and these oospores are generally growing or commonly growing within the structure, that is normal sporangium. Normal sporangium, or you can say also oosporangium. Oosporangium is the specific sac-like structure within which the oospores are generally growing. Last one you can see here is a zygospore, that is commonly found in case of another fungi, that is Zygomycetes or Phycomycetes, and in case of them also the spore sac-like structure which is commonly found, that is the sporangium, that is the normal common spore sac, sporangium. Within those the zygospore is commonly forming. In the next slides we will be discussing about this specific type of sexual mode of reproduction, as well as how the zygospore is actually forming, or how the zygospore structure is related or it is involved in the sexual mode of reproduction, that we will be discussing in the next slide. So I am just going for the next slide. You can see here the various kinds of spores, whatever I have just discussed in the previous slide. You can see here the various kind of spores. What you can see, that is the ascospore. First of all, already I have shown in the previous slide what is ascospore, commonly found in case of ascomycetes fungi. Next, basidiospore is there, which is commonly found in case of basidiomycetes fungi. Oospore is there, that is commonly for oomycetes fungi, and then zygospore, that is for zygomycetes fungi. So these kind of 4 spores are there for sexual mode of reproduction in various different classes of fungi. So I think you have seen the diagram or the pictures of different spores very clearly. Now I am just moving for the next slide, and let's see in the next slide we have to discuss about the mechanism of sexual reproduction, or already I have told you in the previous slide, that is the zygospore, how the zygospore is forming, that one we have to discuss here. So for that reason, first of all we have to know about the word, what is plasmogamy. Plasmogamy means, actually this gamy word you all know, gamy means actually the fusion, okay, or the gametes. From the gametes this gamy word has come. So this plasmogamy means when the cytoplasm of 2 different gametes are fusing with each other. As an example, syngamy. In the syngamy, what you have understood, in the syngamy we all know that is the fusion of gametes. So plasmogamy means when the fusion of cytoplasm of 2 different cell or 2 different rather gametes is happening, then it is known as plasmogamy. So here during the zygospore formation, the 2 gametes, their cytoplasms are fusing with each other, that is plasmogamy. Next, after cytoplasmic fusion, the next type of fusion is happening in between the nucleus of 2 different gametes, obviously male gamete and female gamete. So that specific fusion of the nuclei of the 2 individual is commonly known as karyogamy, because karyon word means nucleus and gamy means fusion. In case of karyogamy, here 2 different words we have to keep it in our mind, their meaning. What are those words? First one dikaryon, and second one is dikaryophase. So what does it mean? First of all, when the karyogamy process is starting, initially both of the nucleus is just present side by side for few minutes. Okay, after that those specific nuclei are fusing with each other. So before fusion, when the nuclei are present actually side by side, that specific structure is known as dikaryon, and in which phase this specific dikaryon structure is found, that is commonly known as dikaryophase. And just the dikaryophase has come, automatically those specific two nuclei are fusing with each other, and after fusion they are forming the next structure for the sexual mode of reproduction, that is the zygospore. So I think the plasmogamy, karyogamy as well as the dikaryon and dikaryophase, these four words is very much clear to each and everyone, and this is the, you can say rather, this is a small, very small flow chart of sexual reproduction in case of fungi. Okay, so I am just going for the next slide. In the next slide you can see the another special feature about the fungi, that is obviously the structural feature, that is hyphae and mycelium. What is hyphae? Hyphae is the branching filament or thread like structure which is commonly found in case of fungi. So in this figure you can see properly, these are the small thread like structures which are actually making the thallus of the fungi. So that is commonly known as hyphae. Hyphae in plural and singular you can consider is as hypha. Another one, mycelium is the mass of hyphae, because the mass of all threads or filaments, when it can be found in a network form, then it is commonly known as mycelium. So fungal hyphae, when they will be getting accumulated more or assembled more and forming the network like structure, then it is known as mycelium. So this is the mycelium structure. You can see it is more condensed and containing so many small minute threads or filaments, those are hyphae, and entire the mass or entire the network which has been formed, that is known as mycelium. So I think this slide also about the hyphae and mycelium is also very much clear to each and everyone. Moving for the next slide. In the next slide you can see there we are discussing about the respiratory system in case of fungi. Here only one thing we have to keep it in our mind, that fungi is showing two kinds of respiration only. One is aerobic respiration. Always we know in the presence of oxygen this kind of respiration has to be performed, and anaerobic respiration, some specific fungi which are parasitic, those are performing or those are capable of anaerobic respiration, because in absence of oxygen also they can respire, they can survive. So during the aerobic respiration we all know that the glycolysis process is happening, and after glycolysis the pyruvic acid we are getting, and if the anaerobic respiration is taking place after pyruvic acid, from their lactic acid fermentation or ethanol fermentation is commonly happening in case of organisms. So in case of fungi also same kind of phenomenon is found. Okay, so this is the respiratory system found in fungi. Moving for the next slide. In the next slide we are discussing about the excretory system in fungi. What about the excretory system? If you see the excretory system properly in fungi, obviously fungi has no solid or liquid waste material, so they have no excretory organ even. So when they don't have any kind of excretory waste product, obviously there is no need to release those from the body. So for that reason the excretory organ, that is also not present properly in case of fungi. So that is the main thing about the excretory system in fungi. So I think we have all understood about the special features of fungi, what we have seen up to this slide. Now we are just moving for the next slide, and there we will be discussing about the classification of the fungi. So let's see what about the classification of the fungi. Here in this slide we can see about the classification of the fungi. First of all, see carefully, the fungi is classified into two different categories based on the presence or absence of septa. Septa means one kind of membrane which is segregating the entire hyphae of the fungi into multiple compartments. So if those compartments are present, or if those septa are present, obviously those are commonly known as septate fungi, and if in case of any fungi those septa are absent, those are commonly known as aseptate or non-septate fungi. Keep it in your mind, those aseptate or non-septate fungi can be also called as phycomycetes, and from the name you can understand phyco word means algae and myco, from that word mycetes have come. So mycetes means fungi, that means this specific fungi is showing the algal features also, or it is looking like algae, so that's why it is commonly known as algal fungi. Now we are going for further classification of aseptate fungi or phycomycetes. Phycomycetes is further differentiated into two different subclasses. One is zygomycetes, another one is mastigomycetes. What is zygomycetes? Zygomycetes is a such kind of fungi which is commonly forming the zygospore. Okay, and by that they are commonly or normally performing the sexual mode of reproduction. Keep it in your mind, these zygomycetes are non-flagellated spore. These zygomycetes are non-flagellated spore, that is spore containing, that we have to keep it in our mind. Mastigomycetes is the another kind of fungi which is containing flagellated motile spore. So zygomycetes is containing non-flagellated non-motile spore like zygospore, and mastigomycetes is producing flagellated motile spore. Okay, so by that specific spore, flagellated motile spore, they are commonly performing the reproduction, mastigomycetes. Another type of the fungi what just we have told about the definition, that is septate fungi. That means in case of which fungi the entire hyphae is subdivided into multiple compartments due to presence of some partitions. So that septate fungi is subdivided into three different categories. First category is ascomycetes. Just we have listened the name of ascomycetes when we have studied about ascospore. So these fungi are commonly known as sac fungi because they are sac-like structure showing. Second one, basidiomycetes. When we have discussed about the basidiospore, there we have just listen this name. Basidiomycetes is a club fungi, that means club shaped fungi, and last one is a deuteromycetes. What is about that? Deuteromycetes is commonly known as fungi imperfecti because it is not perfect fungi due to some presence of some specific features. So for this specific classification of the septate fungi, I think you have all understood. So this is the overall classification of the fungi, where first the fungi is subdivided into septate and aseptate form. Septate is classified into three different categories: ascomycetes, basidiomycetes and deuteromycetes, and aseptate is subdifferentiated into zygomycetes and mastigomycetes. But in the next slide we will be discussing about the comparative study among these specific different classes. But there we will be discussing the comparative study in between these phycomycetes, ascomycetes, basidiomycetes and deuteromycetes. Phycomycetes we have not segregated into zygomycetes and mastigomycetes in that comparative study. Okay, we will totally take this mastigomycetes plus zygomycetes as a phycomycetes and then remaining these three. Okay, so going for the next slide, let's see. So in this slide we can see here properly the difference in between septate and aseptate fungi. It is written. Septate fungi means where the septa is present. In case of aseptate fungi, obviously septa is absent. In case of septate fungi, multiple compartments are present due to presence of septa or membrane, because it is only differentiating the entire hyphae into multiple compartments. And in case of aseptate fungi, obviously there is a single long compartment entire through the length of the hyphae, because there is no compartmentalization can be found. Next one, in case of septate fungi, each compartment is containing with single nucleus. In the figure you can also see here, in the septate fungi, these small septa which are actually present, due to presence of that, each and every compartment contains only single nucleus. But in case of aseptate fungi, the entire hyphae is considered as a compartment. So for that reason, it is containing multiple nucleus. So that's why they are multinucleate, or it can be also called as cenocytic hyphae. Cenocytic hyphae word means containing multiple nucleus throughout the entire length of the hyphae. Okay, going for the next slide. In the next slide you can see about the comparative study among fungi. In the comparative study among fungi, we can see here 4 different classes we have written: phycomycetes, ascomycetes, basidiomycetes and deuteromycetes, with the features are also written. So one by one feature we will be just discussing, and in different classes of fungi, whatever the variations are present in their features, that we can see also. First one, that is a common name. Common name of the phycomycetes we all know, that is the algal fungi. In case of ascomycetes, obviously that is known as sac fungi. In case of basidiomycetes, that is known as club fungi in a common, and deuteromycetes is known as fungi imperfecti or imperfect fungi. Next, if you consider habitat, you will see that in case of phycomycetes or algal fungi, they are commonly present in aquatic body. They can be present in, or they can be growing on decaying wood, as well as they can grow in damp places, shady damp places. In case of ascomycetes, you can see them on the soil surface, as well as they can float on the water body or aquatic body. In case of basidiomycetes, if you see, they are commonly found in soil. They can commonly grows on the log or tree body surface. Log means that is a dead plant body or plant stem. So on that log, on the tree body, existing plant body, this specific type of fungi can survive. And deuteromycetes, which is fungi imperfecti, that can be present in aquatic body. Okay. Next, Based on the presence or absence of septa, we have differentiated them. We all know that Phycomycetes is the aseptate fungi because the septa is absent totally. But if you consider Ascomycetes, Basidiomycetes and Deuteromycetes, you can see the septa is present. So for them all, I have written present. Now the number of nucleus. Based on the number of nucleus also, obviously they are different from each other. Already we know in case of Phycomycetes, septa is absent. That means they are multinucleated, or they can be, in one word, they can be called as sinocytic. But in case of Ascomycetes, Basidiomycetes and Deuteromycetes, they are all containing multiple compartments, and each and every compartment containing single nucleus. That's why they are all uninucleated. Okay, going for the next. Here also some specific points are written. Next point is that asexual reproduction. In case of asexual reproduction, keep it in your mind, Phycomycetes are producing zoospore and aplanospore, and Ascomycetes are producing conidia, and that conidia is present on the sac-like structure or the stalk-like structure, that is conidiophore, as well as on the surface of mycelium. These conidia are present, which is commonly found in penicillium also. In case of Basidiomycetes, asexual spore cannot be found. They are vegetatively reproducing, and that is by the process of fragmentation. Next, in case of Deuteromycetes, asexual mode and vegetative mode of reproduction both can be found. And in the vegetative mode of reproduction, obviously the budding process found. If you move for the sexual mode of reproduction, you can see in case of Phycomycetes, they are forming zygospore, and the formation of zygospore by the plasmogamy and karyogamy, that already we have discussed in the previous slide. In the Ascomycetes, you can see they have ascospore, which is functioning as a sexual structure, and that is forming within the ascus. Already that we have studied. In case of Basidiomycetes, they are forming basidiospore, and that is also forming within the sac-like structure, that is basidium, that is a fruiting body also. And in case of Deuteromycetes, obviously the sexual spore is absent. Now the point is coming, in case of Deuteromycetes, only asexual and vegetative reproduction is happening, but the sexual mode of reproduction, like the other fungi, it cannot be found. That's why Deuteromycetes can be called as, or commonly known as, fungi imperfecti. So previously I have just told this term fungi imperfecti, but why it is imperfect, that is discussed in this point, that is a sexual spore absent. That's why they can't perform the sexual reproduction. So they can only perform asexual reproduction. That's why they are fungi imperfecti. Next, fruiting body. Obviously in case of Phycomycetes, the fruiting body is absent, there is no fruiting body, but in case of Ascomycetes, the ascocarp is present as the fruiting body, or ascus, we have also studied that. In case of Basidiomycetes, basidiocarp is the fruiting body or basidium, and in case of Deuteromycetes, that fruiting body is also absent, that cannot be found because they are not producing any sexual spore. So for that reason fruiting body is not necessary, because this is necessary only for the sexual reproduction. Just moving for the next slide, there you can see that we will be discussing about the examples. In case of Phycomycetes, whatever the type of examples, you can see here Saprolegnia, Rhizopus, Mucor, Albugo, Pythium. These are the example, and within that the figure of the Rhizopus is given. You can see Ascomycetes, that is the sac fungi. Within that, which of the fungi are coming? First one yeast, second one Aspergillus, third one Penicillium, fourth one Neurospora crassa, and fifth one it is given Peziza. So within that, Neurospora is the most important type of the fungi because it is utilized in several cases during the research. So for that reason Neurospora figure has been given. If we go for the next type of fungi, that is the Basidiomycetes, that is also known as clove fungi, and it can be found in case of which fungi, Basidiomycetes are which specific fungi? First one Agaricus, Polyporus, Puccinia, Ustilago, Lycoperdon or Lycoperdon. So Lycoperdon as well as including all kind of fungi, these are all coming within the category of clove fungi or Basidiomycetes. And the last one, that is a fungi imperfecti, Deuteromycetes, that is Sarcospora, Calectotrichum, Trichoderma, Pyricularia, Fusarium. So this within this Fusarium figure has been given, and with that also Trichoderma is the known name to us. So these are the total examples of various fungi classes, and with that one representative fungi from each and every category. Their figure has been shown. So I think starting from the kingdom fungi special feature up to this slide, we have all understood properly. I am just trying to show you all the slides again. So we have discussed about the special feature, that is the mode of reproduction here, asexual mode of reproduction. So these are the figure of various kind of spores which are involved in the asexual mode of reproduction. Next we have discussed about the sexual mode of reproduction. Next we have discussed about, or we have also shown the figures of various kind of sexual spores. Next we have seen about the sexual mechanism, sexual reproduction mechanism or zygospore formation by the plasmogamy and karyogamy, and at last the zygospore is forming. Next we have seen what is hyphae and what is mycelium, the structural difference also it is very much clear to us. Respiratory system in fungi, both kind of respiratory system, aerobic and anaerobic, can be found. In this slide we have discussed about the excretory system also. In the next slide we have discussed about the classification of fungi, septate and aseptate fungi. Within septate, ascomycetes, basidiomycetes, deuteromycetes. Within aseptate, zygomycetes and masticomycetes, and aseptate is commonly known as phycomycetes. Septate aseptate fungi difference with the figure, it is very much clear to everyone. Septate fungi means, always keep it in your mind, they will be uninucleated, but aseptate fungi will be multinucleated or cynocytic, because their entire hyphae is considered as a compartment. Next you can see the comparative study among fungi classes based on various features, and in this slide, in the next slide you can see the same thing, because a comparative study has been done, and in the last slide you can see about the examples of various different fungi classes. So I think this entire video, whatever I have made on this kingdom fungi, you have understood properly and you have enjoyed a lot this video. Thank you everyone.